Now we have got time for some questions, if there are any. I know this is quite an in-depth subject. Yes, there's a question in the front there.
I think the capacity for repair, even after many years of illness, it's very good to hear, because one of the depressing things is that when you get transverse myelitis or similar, you read that after a year, what progress you haven't made after a year, you can forget, and that's not been my personal experience. I'm talking to other people that progress does keep going on, and so it's nice to hear that that is a possibility. Going back to the exercise increasing the brain stem cells, do we know that those brain stem cells actually sort of get into the completion of the repair process, or are they just there sitting on your log?
All right, so Charles and I will go back and forth. So the first point is, let me just, I'm not sure it was the question, but let me answer it. It's an important point. Is kind of, in terms of stem cells and the environment into which the stem cells would be placed, is there a time period in which those stem cells could repair, and is there a time period beyond which it's too late? That's a very important question. We don't know the answer. I'd be interested to hear what Charles says on this. You know, I think that there's probably a little bit of a balancing act in terms of timing. So in the throes of an acute inflammatory destructive process, transplanting stem cells, it may be too early. But at the same time, beyond a certain point, I think it's probably going to be too late without reestablishing a local environment that would support the transplantation. Let me get Charles' input on that, and then we'll go to your second one. Oh, you do?
Yeah. No, I agree. Just to talk about exercise for a second, the stem cells, the area of the brain that they were looking at there was the area called hippocampus, which is involved in learning and memory. And I think the current view would be that those cells are generating new neurons involved in our ability to cement long-term memories. That's to some extent speculation, but I think that's probably what's going on there. And I would completely agree. I think it's very clear that there are two quite distinct problems in terms of stem cells and repair. In many cases, the problem is that it's not the lack of stem cells, it's the environment in which they're trying to repair, exactly as you've just said. And we know that in many lesions in patients with MS, which I keep using as an example, I know, but we know that in many lesions contain plenty of stem cells, they just can't complete the repair process. But equally, there are lesions where there simply aren't enough stem cells. So I think there is probably value both in approaches to increase the number of stem cells and also to deal with the environmental problems.
There's a series of experiments that kind of illustrate this point. So a bunch of very clever scientists have induced a peripheral nerve injury where they literally cut the peripheral nerve, and if they reestablish the two ends together, the axons will regrow and repair will be perfect. If, by contrast, you keep them apart for 6 months and then put them back together, it's too late. But you can do something clever, where if you induce one injury, you keep them apart for 6 months, and another nerve is acutely injured, you can now transfer the two, and you will get regeneration to occur. The reason why that's important is because it says the environment tuckers out after a while and would not support axons regrowing or remyelination. But if you can wake up that local environment, or if you can change that local environment, you can actually turn back the clock, so to speak, where you now really can induce a regeneration. So that's going to be a key thing as we go further in transplantation or stem cell therapy for those who are further out from an acute injury.
Another question. Yes, just behind the lady with the gentleman with the blue pullover.
Thank you. I must say, a fascinating discussion this afternoon. Really enjoyed it. Thank you. There were two aspects, and forgive me not being technical here, but in terms of suppressing the lingo element, and also in stimulating the RXR gamma gene, are these mutually exclusive piece of works that are going on? Are they combined? And what is the combination of work between what's going on in the States and what's going on in the UK?
Yeah. That, by the way, was very technical. You apologize. But I'm afraid I can't accept your apology. It's quite technical. No, I think ultimately, I think we all believe that no one therapy is going to do this, and that it's really going to take a combination therapy to really induce maximal repair. And I think these would be beautifully synergistic therapies together. You want to talk about the...
Yeah, I completely agree with that. The vision would be that patients with these complex diseases will take a cocktail of drugs, just as patients with other complex diseases are well used to taking drug cocktails. So for MS, for example, I could see patients taking drugs to prevent the inflammation, drugs to protect the axons that are there, that still haven't died, and drugs to promote regeneration, and probably a drug to deal with the side effects of one of the other three as well. Any effective drug will certainly have side effects. And you know, there are plenty of other examples of other areas of medicine where this is happening already, so I don't think there's any difficulty there. You asked a very interesting question about collaboration. And the collaborations between the UK and the US, and in fact Europe and the US, is very strong. In fact, we were all involved in a very important campaign funded by the National Multiple Sclerosis Society in the US called Promise 2010. And that involved groups in the US, groups in the UK, and groups really all over Europe and Canada as well. Brought them all together, and there's a lot of collaborations that have come out of that. So no, there's no barrier.
Final part, if I may, is just how far are we away?
Yeah, I would just say that you all should know that the UK really has, I think, led the world in terms of understanding the niche biology or what we need to know about stem cells and their differentiation. And that which Charles described to you is just brilliant. I mean, that synergy that you would get from putting all of these people together in that design is spectacular, and there are not many centers that have that much vision to put those groups together, and I think that will very much accelerate things. You are starting to see stem cell trials now. So some are underway in, for example, ALS, motor neuron disease. There's a trial underway. In traumatic spinal cord injury, there is one which is underway. So you're beginning to see those, but these are very early stage. These are what we call phase 1 studies, and they really, at best, will give you a sense of the safety of the approach. Really will not in any way get you a sense for efficacies to whether they're working. You know, and I've told my patients, I don't think you want to be in a first-generation stem cell trial. I think you want to be in the second generation or third generation stem cell trial. When we figured out how to deliver them, we really know what level of immunosuppression, if any, do we need so that we can protect the stem cells from being rejected, but not put the patient at too much risk from an infection. Those types of things are really important issues that have not yet been worked out.
And just to point out, the early trials are happening, and they're happening both for the immunomodulation, usually using a stem cell called a mesenchymal stem cell, and there have been 2 MS trials in that area, but also for cell replacement, and the spinal cord trial would be a good example of where they're actually trying to replace a cell that they think will promote spinal cord regeneration. One of the problems with these trials, though, is they're very, very small numbers of patients. And those patients have to be chosen with enormous care so that they have exactly the clinical picture that enables the researchers to answer the questions that they're setting out to ask, that they're amenable to the investigative techniques that they have. And I know that people find it intensely frustrating because we get letters every day saying, you know, can I be in a clinical trial? And the answer at this stage, I'm afraid, is no, not because we don't wish it, but because the trials are very small and we have to be incredibly selective about who we choose.
Another question. Yes, the lady in the middle, halfway back. Yeah, just by you there, that's it.
Hello, I have an which is in repair and relapsing.
Are you switched? Is that switched on now?
I don't think it's working.
There we go. On your right. There we go.
Okay, so I have an MO, and because it's relapsing, you know, could stem cells ever be used? You know, do they have sell by date? Do they last only so long? Could you have a bank somewhere inside you of stem cells so that rather than using them to repair, well, you would still be using them to be as repair, but as the damage happens, if you were having a relapse and as the damage happens, they would be right there behind, you know, to repair straight away, and so you haven't got any delay. Would that, do you think, ever be a possibility?
So the point that you're worried about is that with NMO, since you have recurrent attacks, that any stem cells that might have been used initially would be destroyed by a subsequent attack.
Uh-huh.
And that's true, although in neuromyelitis optica, you know, the regions that of the spinal cord differ in many cases between attacks. But I think the point you're raising is, can you kind of have a cell bank that could be used for multiple uses after multiple attacks?
Mm-hmm.
Yeah, so I think that's possible. I mean, I think what a lot of people think is going to be possible is ultimately an autologous transplantation paradigm, where if we can generate cells that have the potential that we wish for a particular disorder, we can take them from skin, potentially from you, do what we need to do to generate the stem cell population, for example, for remyelination, expand that population and freeze it away. And ultimately could use it, you know, multiple times if we need. I think that's possible. It's a long way off. But I think autologous expansion of stem cells to be used, you know, potentially multiple times is a reality. I think, though, with NMO, as with multiple sclerosis, the real hope is that it's an appropriate therapy that halts further attacks while repairing the injury from the attacks that have occurred to begin with. So that hopefully you can minimize the chance that the transplanted cells will be attacked themselves.
Thank you.
I think, ladies and gentlemen, at that point we'll draw stumps, as they say. Thank you very much to our two speakers for an update on stem cell research. It's now 3:10. We're due back in here at about 3:30, so time for a quick cup of coffee, and then I'll see you later for the rest of the afternoon programme.